So, Dr. Isabel Emanuel is an American Association for the Advancement of Science and Technology Policy Fellow at the United States Botanic Garden, where she is developing a framework for research partnerships with academic, federal, and non government organizations and support science education and outreach initiatives. Prior to starting her fellowship, Isabel earned her PhD in plant pathology and has robust research experience in plant disease diagnostics, agricultural security, and microbiological food safety. Welcome, Dr. Emanuel.
Thanks, Courtney, for the great introduction. And thank you, everyone, audience, speakers, advisors, for joining us today during your lunch hour. For those of you tuning in again, you are here for the first session. Please give yourself a shout out in the chat. Would love to see who's here from last time. So today's program is, again, our second and final meeting of Research Lab Look-In for the summer, and it's going to focus on plant defense and competition strategies. And so both of our speakers today are graduate students who are actively conducting research in these areas, and they're going to be breaking down their research concepts and approaches so that we can all better understand how plant science questions are being answered in the lab today. Will Parrot is joining us today from Virginia Tech, where he actually began his academic journey as a computer science major. So Will discovered his passion for ecology after taking an elective course during his undergrad at Virginia Tech, and now, after years of study, is currently wrapping up his first year as a graduate student in Dr. Carrie Fear's Forest Ecology Lab. So today, Will is going to be discussing his research involving the development of tools to monitor defense chemicals in trees and allow us to predict resistance of these trees to invasive insect pests. So welcome, Will.
Hi, thank you very much, Dr. Emanuel, for having me, and thank you all for joining us today. We have a really cool session in store for you guys. Let me just get this presentation up. All right, so today we're going to be talking about plant defenses, very broadly going into how plants can defend themselves from insect pests. And then we're going to talk a bit about our research at the Frazier lab in Frazier fir systems with relation to the balsam woolly adelgid. So just to give you guys your bearings, here's a brief overview of how our presentation is going to look today. So we're going to start off talking about plant defenses. Why do plants need to defend themselves? How do they do it? And why do we care about looking at the resistance of these different organisms? Then we're going to go more specifically into the Frazier fir system and how it's been impacted by infestations by the balsam woolly adelgid. And then we're going to break down our research that we're doing into this system and how we're hoping to contribute to management practices in the long run. And then after all of that, as Dr. Emanuel said, we're going to open the floor up for questions. So feel free to put them in the chat as we go. So without further ado, let's get into it. So you may be wondering, why do plants need defenses? You know, they can be pretty innocuous. They can blend into our background, but plants are really versatile organisms. So they have certain characteristics that predispose them to predation or herbivores. So the first of these characteristics is that they are sessile. As they grow, they stay in one place, and they do this throughout the course of their life. And while this provides a lot of different advantages, it also opens them up to risks from environmental threats and from other organisms that want to feed on them. In other words, plants stay still, but the world keeps moving around them, and they need to adjust themselves accordingly so they can continue to survive and reproduce. The other characteristic of plants that predisposes them to issues from herbivores is that they are considered primary producers. So unlike us, who need to eat other things in order to make energy, plants are able to produce their own energy through photosynthesis, collecting energy from the sun and turning that into their own resources. Unfortunately, that makes them delicious for other organisms to come in and highly nutritious for them to eat For instance, fungi, especially in forests, fungi and other microbes, and what we'll be focusing on primarily in this presentation, insects. So, long story short, plants are sitting ducks, and they need to be able to develop an arsenal of defenses that allows them to continue to survive in the face of so many threats. So how do they do this? Well, there are two main categories of defense within a plant's arsenal that they can use to fight off these threats. And the first of these, and perhaps the most straightforward, is with mechanical defenses. Now, mechanical defenses are those structures that plants put up to prevent organisms from feeding on them, or to deter organisms from continuing to feed. For instance, with thick bark, some trees will have thick bark on them that prevents certain beetles from being able to bore into the side. Or they can have deterrents, such as crystals that form between cells that actually irritate different herbivores that are passing through the tissue of the plant. But as I said, these are only half of the battle here. There's a more subtle set of defenses that we like to focus on in the Führer lab that also contribute significantly to the protection of plants against herbivores. And these are our chemical defenses. There are a lot of different classes of chemicals and things I'm not going to really get into in this presentation. We just don't have the time. But very broadly, chemical defenses can fit into one of three categories. The first of these categories is chemicals that act as deterrents to insect pests. So imagine you're an insect. You're going and you're looking for some food. You see two flowers in front of you, but one of the flowers just smells awful. It's super pungent. It just does not smell good. Or picture you're taking a bite into a plant and it's super bitter. You're probably not going to continue to feed on there. Well, that's chemical defenses. Some of these chemical defenses push away certain herbivores to different organisms around them. On the flip side, there are actually chemicals that can act as attractants. So when a certain type of pest feeds on a certain type of plant for long enough, the plant will exude a chemical regularly that certain predators of the pest can actually pick up and use to feed on it. What this does is it creates a mutualistic relationship where the predator of the pest gets food if it hangs around the plant, and the plant gets protection from the insect pest. It's a very indirect way of doing things, but it's still very cool. That's not to say, though, that plants can't defend themselves directly through toxins. As the name suggests, toxins are chemicals that plants can accumulate within their tissues in order to directly harm or even outright kill certain insects that are feeding on them. For instance, some oak leaves have chemicals within them that protect them from caterpillars. So this is all well and good, but how do they employ these different defenses? Well, there are multiple ways they do this as well. They can employ them as passive defenses or active defenses. So passive defenses, also known as constitutive defenses, are those that a plant produce as it grows without the pest necessarily having to be there. And it's essentially setting aside resources so that if something comes in and starts feeding on the plant, the defenses are already there. Think of it like insurance. You set that money aside in case anything happens. You may not need it at the moment, but when you do need it, it's there. Another way you can think about it is with people. So people actually also have different types of defenses. And a good passive defense that we people have is with skin. Skin is a passive defense. It doesn't necessarily feed into pumping blood throughout our body or letting us think or letting us breathe, but it does protect us from environmental irritants like ultraviolet light or dust. On the flip side, they're not just sitting ducks, these plants. They can also respond to attacks from pests. And these are what we call active or induced defenses. For instance, the picture that we have here under this category is a process known as compartmentalization. When a fungus or some sort of decaying agent enters a tree, that tree can actually block off the cells around it to keep it from spreading further into its tissue. To go back to our human analogy, this is our fevers. They make us, it doesn't feel good to have a fever, but what the body is doing is responding to those threats. It's responding to whatever invading pathogen is causing us to get sick. So all these concepts of mechanical and chemical defenses, passive and active defenses, if you take a step back, they play into a larger categorization that we can take for different plants. And one that we are very focused on in our lab, and this is resistance. So put simply, the resistance of a plant to an invading pest or pathogen is its ability to withstand that attack and continue to grow normally. And this different plants will fall along a spectrum that we have here. So on the far left end of our spectrum, we have susceptible plants. These are ones that don't have any defenses. It's wide open to attack, it's wide open to feeding, and usually susceptible plants will die to a specific pest or pathogen if they're susceptible to it. On the flip side of that, we have resistant plants. Resistant plants are those that can not only fully withstand an attack, but continue to grow normally and actually deter whatever pest or pathogen was impacting them. And then somewhere in the middle, we have tolerance. Now, you may be sitting here wondering, why is this important? Why do we need to think about this? Well, it's important in a wide range of contexts, from agriculture to plant breeding to conservation of natural resources. And I think the best example of this is with the American chestnut. The American chestnut was a very significant tree throughout eastern hardwood forests leading up to the 1900s. It was very good for providing food to people and other wildlife. And it also had a lot of cultural significance. But unfortunately, the American chestnut was decimated when a fungus called the chestnut blight came through and wiped out the forest. And it's a tragic story. This was a wonderful tree that we had, but we can learn a lot from it. If we had the ability to study the resistance of chestnut and maybe cultivate more resistant trees, it's possible that we could have still had this tree within our landscape. Now, this is important to consider within a broader context. We need to study these trees while we have them, especially those that are threatened and endangered, such as the Frazier fir, which is the main tree of study within our research. So the Frazier fir is most commonly known as a Christmas tree. It's got a very nice form. It's planted very widely in Virginia and North Carolina for the commercial Christmas tree industry. But few people actually know that it's an endangered species within its own range down in the Great Smoky Mountains and Mount Mitchell and Mount Rogers. It only exists on a few mountaintop peaks as a holdover from the last time glaciers were down in that area. As the glaciers pulled up, Frazier fir alongside red spruce started to grow, and they dominated the peaks for thousands of years, until climatic changes and logging practices in the early 1900s pushed them to the top of mountains where they are now. And they can't really spread on their own past this because the climate is not right for it. As such, it's important to conserve the natural populations that we have, especially in the face of invasive pests such as the balsam woolly adelgid. So that picture on the left is a close-up picture of the adelgid. They're very, very small. And they have these white, waxy, almost hair-like substances on their back. And they have a long, straw-like mouth that they can feed through the cells of a Frazier fir to get to where they store sugars and starches, also known as the parenchyma. And this pest was introduced to the US in the early 1900s, but it was found in Frazier fir's range in 1956. One of the problems with it is as it feeds, one, they feed en masse. That picture on the bottom there is a strong example of a Frazier fir that's been infested by the balsam adelgid. So you have all of these sugars being taken out of the tree, but you also have the fact that as the adelgid feeds, it's actually exuding a saliva that causes abnormal growths like we see on the top there. That's called gouting, which can cause defoliation and poor form, which is more important to Christmas tree growers. But it can also cause the tree to be unable to transport water enough to sustain itself. And unfortunately, between 1956 and the 1980s, one study suggests that about 66% of the mature Frazier fir within their native range had succumbed to infestation by the adelgid. And just to put that in context, picture that group of trees on the right as a mature Frazier fir stand before infestation by the adelgid. And this is what it would look like after. It is a significant loss. So there's a lot of interest in finding trees that can tolerate infestation and finding ways to promote those trees in the landscape. So how might Frazier fir be able to resist this? What would we find in a tolerant versus intolerant cultivar? Well, there are a couple of different things they can do. Back to those constitutive and induced defenses. So in terms of constitutive defenses, those parenchyma, the cells in Frazier fir bark that store all the sugars and the energy, they may sometimes be laced with defensive chemicals that can deter the adelgid from feeding. In addition, and this is a pretty common thing to see in other conifers like pine or spruce, is the production and storage of resin in specialized ducts throughout the tree. Now, resin is a very special substance that can physically trap insects if they're boring in, but they also contain, resin also contains a lot of different defensive chemicals that can help the tree respond. In addition, a tolerant fir would be able to make its bark thicker, which would prevent the straw-like mouth from being able to reach those starch cells as parenchyma. And they can increase production of that resin. So there are things that we would expect to see, and that's a bit what our lab is trying to capitalize on in our project. So what we're working on right now is seeing if we can tell what fir are tolerant or intolerant to the balsam woolly adelgid based on data from a near-infrared scanner. There's a lot to go into with near-infrared scanning, but very broadly, near-infrared spectroscopy can detect chemical differences within plant tissue with very limited impacts on the tree in the long run. So that's what we're applying here. We went out to an area where we knew the tolerance levels of the trees. They're clonal varieties of trees that were collected from the native range. And we took this scanner, which you can see on the left here, and scanned the needles and the bark of the tree. This is our lovely lab mates out in the field actually doing this collection here. And what we can do with that data is we can put it into a machine learning algorithm, and that will tell us whether there is a significant difference between the tolerant and intolerant trees based solely on that near-infrared data. It's a bit to wrap your head around at first, but it's a really cool process in the long run. And what we found so far is that these models that we're putting the data into are actually able to detect the difference between the tolerant and intolerant fur. Depending on the tissue that we scanned, we found that the bark tends to have higher accuracies in being classified as tolerant versus intolerant than the needles, which makes sense because, as we talked about before, we would expect the defenses for the Frazier fir against the balsam woolly adelgid to be contained within the bark. In addition, these models that we're using, they take in all of the wavelengths that our near-infrared scanner collected, and it sorts through them, and they actually spit out which ones were the most important in determining whether a tree was tolerant or intolerant within the machine. And what we're finding is that there are wavelengths here that show up pretty consistently across all of our models. This points us in the direction of, oh, there is a certain chemical feature within the bark or within the tissues of this plant that is contributing to its tolerance against the adelgid. And that's one of our next steps, is actually extracting the chemicals from the needles in the bark and using those to feed back into our sensor in order to be able to go out into the field and tell, without tearing down a tree or sending off to a lab for processing, whether it would be able to withstand an infestation by the adelgid. So that's one of our next steps. And then after we've verified that this is possible and that we can do this relatively reliably, we're going to see if we can couple our tolerant measures with environmental variables like humidity or temperature to see if that's playing a role into the ability of the adelgid to resist infestation. But these are just our next steps for now. That's about all I have to say on our project. Thank you all very much again for coming out and learning from us, and for our organizers for hosting us. If you'd like to reach out with further questions, you're welcome to ask them now, or if you would like to, you can reach out to my email. And I'd just like to acknowledge our funding sources and all the people that made this project possible. Thank you all very much. And with that, I'll take questions.
Awesome, Will. Thank you so much. That was really interesting. Wow, it seems like this is a pretty devastating pathogen or pest. I'm so used to pathogens. How quickly do these symptoms progress from an infestation? So you showed, I think, over half of the trees can be wiped out in a Christmas tree farm like this. So how quickly does that progress? And is there anything that growers in these areas can do right now?
So the symptoms for this, it depends on the tree, right? Because some trees are already going to have some stressors. They're already going to be kind of sick. But a lot of the studies that have come out have suggested that a full infestation, like we saw in that earlier slide, can kill a tree between 2 and 9 years without any management. As for what that management is, it's pretty hard to say. So I could not tell you off the top of my head right now. But.
Yeah. And when we are looking at plants that are used for ornamental purposes, the tolerance for symptoms from diseases and pests are quite low. So it might actually be faster than complete tree death before that tree is unsellable.
Absolutely.
For those growers. Yeah.
Absolutely. And that's why this work matters so much. We want to be able to pick out what's tolerant or intolerant so we can promote that within our nurseries.
Yeah. Not only for forest health, but also, save Christmas,
Yes. Yeah.
So for those who celebrate, so once you have found trees that have suspected resistance, how do you go about proving this? And then what are the next steps for building upon this resistance to strengthen native forests, or then also create more resistant cultivars for growers?
That's a good question. It's a loaded
Yeah.
question.
Yeah,
So,
maybe too big of a question, Could be.
no, but so I'm not working directly on the plant breeding side of things.
Mhm.
So there is a cooperative at North Carolina State University that we're working with,
Mhm.
known as the Christmas Tree Genetics Program. It's headed up by Dr. Justin Whitehill, and he did different tests on certain. He collected different seeds from the ranges that were impacted, and then exposed them to the adelgid and saw how they responded in the long run.
Mhm.
That's very, very broadly what you would see in testing for that resistance.
Yeah, just kind of like outdoor trials confirming things. That's usually how these tend to go. So that makes sense. But for Christmas trees, that would take a long time. So
Absolutely.
yeah. So I know you have some prior industry experience in arboriculture at Bartlett Tree Experts, but now that you're midway through your first year of summer field research, how is it different and what's your favorite part of this type of work?
Ooh, okay. So it's different in that I can take more breaks, which is nice. But no, in seriousness, it's just looking at different aspects of the trees. That's really what this is we're looking, we care about plant health in both contexts, but within arboriculture, it's very much about the long-term sustainment of plants within our urban landscapes. Versus this work, it can be applied.
Yeah,
Sorry.
you're fine.
Sorry, I got lost in
Yeah.
thought there. I guess it's really the application of it would be the
Yeah.
big difference, right?
Yeah.
The work that we're doing here is more applicable to the nurseries and to the land managers actually working within the trees in situ.
Yeah.
Versus arboriculture, we are still working with the land managers, the urban foresters, and that sort of thing. But it's more of a direct application, if that makes sense.
Yeah. And touching a bit upon the applicability of this, we have an audience question asking how what you're doing with these scans can transfer to resistance in a tree, different types of trees and different types of pests and pathogens. So is this specific to this tree and this pathogen, or can this be applied to other hosts and pests?
So that's a really good question. It can definitely be applied outside of just the Frazier fir and balsam woolly adelgid system. As a matter of fact, there have been a couple of different studies preceding this, including one by my advisor, Dr. Führer, which looked at beech leaf disease, which is an emerging foliar pathogen, and using this technology to detect when that disease was starting up before the visual symptoms started. And I guess it really does depend on what tree you're looking at, what defenses might be present, and all of that. But it can be applied. And our hope here is that we can create sort of a framework that can be applied to different disease and pest systems.
Yeah, absolutely. I can see how that kind of rapid screening could be really beneficial, especially when it takes so long to grow trees and screen them and all of this. So really, really interesting stuff, really important. So thank you so much for sharing. Okay, so we are about halfway, so we're going to move on to our second speaker. Abby Koman is an outdoor enthusiast who has always been intrigued by and bothered by the tendency of invasive plants to seize control of native forests. So inspired to take action by a botany course during her undergraduate studies, Abby is now wrapping up her first year as a graduate student in Dr. Oscar Roach's Conservation Biology Lab at Kent State University in Kent, Ohio. And today, Abby is going to be discussing her research, which is dedicated to exploring mechanisms utilized by these invasive plants to outcompete native plants and how this phenomena can harm ecosystems. So welcome, Abby.
Thank you so much, Isabel. Let me get my presentation up here.
Alrighty. So hi, everybody. Like Isabel said, I am Abby Koman. I'm a PhD student at Kent State University in Kent, Ohio. So I study a plant called multiflora rose, which I'm very excited to get to teach you about today. So in this talk, I'm going to cover what is multiflora rose, how it competes against other plants, and how it defends itself. And then I will dive into my own research studying plant pollinator interactions in the Cuyahoga Valley National Park in Peninsula, Ohio, and a couple other sites. So thank you everybody for coming, and let's get started. So what are invasive species? So humans are always bringing and moving plants around the world. And most of the time, this doesn't create invasive species, but sometimes plants will become established in these new areas and they can outcompete native plants, often for light, water, nutrients, or space. And these are considered invasive species. They can cause environmental and economic damage that is incredibly expensive. In the last 60 years alone, invasive species have cost the United States $4.5 trillion in management efforts and damages to agriculture. So as you can see, this is a big issue, which leads me into the specific invasive species we will learn about today called multiflora rose. So what is multiflora rose? It is a type of wild rose that was brought to the United States in 1866 to use for grafting rootstocks. It's one of the most invasive species in the United States. So rose farmers thought that the roots of multiflora rose were really resilient and hardy. So they would cut the roots off of whatever ornamental rose they were trying to grow, and they would graft it with the roots of multiflora rose. Later in the 1930s, people used it for erosion control on hillsides and for fences for livestock because of its ability to form these dense thickets that even cattle cannot get through, which is the photo there on the top. So that's why they are known as living fences. The rose quickly expanded out of control and it's overtaken forests and their edges, open fields, and disturbed land, which can lower property values and destroy land for agriculture. Today, its range expands through most of the United States, except for the Rocky Mountains and the deserts of Nevada. And in many states, it's actually illegal to sell or plant multiflora rose because of this, especially in Ohio, which is where I am right now. So today, let's get to the theme about multiflora rose's competitive strategies. We first need to cover generally, how do plants compete? So all plants compete with each other more or less. Sometimes it's within the same population. So plants of the same species are competing with each other. For example, two multiflora rose plants side by side competing with each other. So this is called intra-specific competition. Other times, plants of different species can compete. So you can have multiflora rose and native Allegheny blackberry side by side competing. So this is called interspecific competition. But regardless of the type of competition, plants can still fight each other for things like water, nutrients, space, and light. Additionally, plants can compete by growing faster than others, which can cast shade on competitors. And if the competitor plants can't get enough light for photosynthesis, they can't make their own food and they will die. So also, some plants can compete by releasing chemicals into the soil, which can restrict or kill other plants surrounding them. And this is called allelopathy. So a good example of this are black walnut trees. They can release chemicals into the soil that can kill nearby sensitive plants and effectively reduce competition. So in the context of multiflora rose, how does multiflora rose compete? Well, the reason why they are so successful at being invasive is because they have something called a dual reproductive system. Essentially, they reproduce in two different ways at the same time. The first is called asexual reproduction, which is basically forming clones of itself. A shoot will form off the stem. It comes into contact with the ground. It will grow roots and become its own plant that is a clone of the parent.
So asexual reproduction allows the roses to get really big really fast. And it can form these dense thickets that can choke out animals and other native plants, especially if it shades out the native plants too much, which can ultimately alter habitats. And let's take a look at this in the context of my own research. So here is one of my field sites, which is the Cuyahoga Valley National Park in Peninsula, Ohio. Here on the left, we have what used to be a farm field. In 2019, the parks decided to turn this into a natural area, and they left it hopefully to return to a grassland or a prairie. But within just 4 years, the entire field became infested with multiflora rose. This is a satellite image from last year in the winter on the right here, showing just how bad this infestation has become in the last 5 years. Each dark spot is a multiflora rose plant, which you can see is covering most of the field. And this really highlights just how asexual reproduction has allowed this plant to take over. The second half of the reproductive system is called sexual reproduction, which focuses on the production of flowers, which get pollinated and turn into rose hips, which are the fruit of multiflora rose. Unlike cloning, which only has the genes of one parent, sexual reproduction comes from 2 different multiflora rose flowers, which can either be on the same plant or on different plants. So this allows for genetically diverse offspring, which is better for the plant overall. It can help the plant bounce back from environmental challenges, such as changes in the habitat or a new disease. And some multiflora rose plants can produce up to 500,000 seeds per year, which is a ton. That you can see in that photo I took there, probably has close to that amount. And these seeds are a huge problem because they can last up to 10 to 20 years in the soil, which is really hard to get rid of. And it has the birds, white-tailed deer, rabbits, and mice will eat the hips and spread the seeds to new locations, which can make it very beneficial for multiflora rose, and ultimately why it's such a successful invasive. So another reason why it's so successful is because it has something called timing strategies that allow it to outcompete native plants. For instance, multiflora rose, it grows leaves earlier in the spring than native counterparts, which allows it to start photosynthesizing faster so it has more resources and a leg up. It can effectively shade out the plants beneath it for a competitive advantage. Additionally, multiflora rose stays green later than native plants, and it holds on to its rose hips throughout the winter when food is often scarce, so animals can keep eating and spreading the seeds throughout the winter. It also does something called mass flowering. So it blooms heavily all at once in a 2 to 3 week period between May and June, at least here in Ohio, with continuous flowering to attract pollinators through both sight and smell. And this essentially can allow it to steal pollinators from native plants. So multiflora rose attracts most of its pollinators by the color of its anthers, which are the male reproductive part of the plant. And the bright yellow color draws in bees and it boosts pollination. Inside the middle of the anthers, you can see there is the female reproductive part called the stigma. And when pollinators come to the plant to feed on the pollen off the anthers, it just happens to brush up against the stigma, hopefully with another multiflora rose's pollen, which will effectively pollinate it. So how does multiflora rose defend? So when we think of defenses, we often imagine animals fighting or fleeing, but plants can't really move, so they have evolved defense mechanisms to protect themselves from herbivores and environmental disturbances. One physical defense are thorns. And a fun fact is that roses don't actually have thorns. Instead, they have what are called prickles. So prickles are outgrowths that come from the epidermis or the skin of the plant, while thorns are modified stems. Either way, they are extremely painful. I've definitely experienced that this summer. And the prickles will deter animals from eating them and getting through the plant. If you look at the photo there, you can see that the prickles they hook backwards and they allow multiflora rose to climb on top of other plants and spread to new locations and shade it out in that way too. Additionally, the dense thickets will shield the inner stems and the roots from grazing animals and can also protect the plant. And lastly, if the plant is cut, grazed, or burned, if somebody's trying to get rid of it, it can produce shoots asexually and effectively resprout and come back, which makes it really hard to kill. Okay, so now we are on to more of my summer research project. And the question I am investigating is, does pollinator access and body size affect multiflora rose's reproductive success? To test this, I used 3 sites in Northeast Ohio. The first was the Cuyahoga Valley National Park, like you saw earlier. The second was a local county park called Quail Hollow in Hartville, Ohio. And the third was a Kent State property called Jennings Woods in Ravenna. These sites all had different mix of habitats of forest, forest edge, open field, prairie, and swamp. And at each site, I selected 10 multiflora rose plants to experiment on. So you can see here my design. Each plant had 5 different types of mesh treatments on them that excluded different sized pollinators. So the first was the control, which had no mesh on it, allowed for full pollination. The second was the 10 millimeter mesh, which is the second from the left there. This excluded large pollinators, such as butterflies and birds, from getting through. Next was a 5 millimeter mesh, so the holes are getting smaller here. And this excluded bumblebees, carpenter bees, and allowed for more medium-sized pollinators. Next, I had a 3 millimeter mesh, which excluded even smaller pollinators like honeybees, but allowed for smaller syrphid flies, and ants to pass through. And lastly, on the right, I have a 1 millimeter mesh, which effectively excluded all pollinators. And the goal here is to see how excluding different size pollinators affects the fruit that is produced within the meshes. So the things I am measuring will show the amount of sexual reproductive success. And to do this, I'm counting the stigmas, which are the female reproductive part, from inside each mesh. And I'm using a scanning electron microscope to count the number of pollen grains on the surface of the stigma to measure pollen deposition. And this will help me show that there are differences in the pollination between the different types of meshes. And in the fall, I will go out to all my sites and collect all the rose hips produced in each mesh. I will cut them all open, count the number of seeds, and I will weigh them. And what we're trying to do here is see if there's a difference in the fruit production or seed amount or weight between the different mesh types. So what do I expect to find? My hypothesis is that the meshes that allow for bigger pollinators will be more pollinated and have more seeds. And the meshes with the least amount of pollinator access, so that 1 millimeter mesh in the photo there, they will have the lowest reproductive success. So my experiment also included some pollinator observations. I wanted to get a sense of what pollinators were getting through the meshes and create a list of actual pollinators of multiflora rose. I identified visitors to species if it was possible. And what I found was that the main pollinators are margin calligraphers, which you can see in that bottom left photo. They are a type of syrphid fly. And this was the most common insect I observed across all 3 of my sites. The second most common are soldier beetles that you can see in the right photo there. These often fell asleep in the flowers overnight and would wake up at 7 a.m. and would start munching away at the anthers. And it was really fun to watch them feast. Other notable visitors that were important were sweat bees, honeybees, which exclusively foraged on only the yellow anthered flowers. So something important to note here is that multiflora rose anthers turn from yellow to brown as the flower ages within a couple days. So I observed that bees do not forage on the brown anthered flowers. You can see on the right, they would not visit that flower. It was pretty interesting to discover. And other pollinators that I observed were many different types of flies, beetles, and ants as well. So other insect sampling, I did 2 other methods. The first was malaise traps, which you can see in that big tent-like structure there. It basically captures flying insects. So the insects will fly into the tent wall, climb up the side and into a tube, which collects them and kills them in a solution of ethanol, which is a type of alcohol. And I sampled these once a week for the 3 weeks that the multiflora rose was blooming so that I could get an understanding of what pollinators or insects in general are at each site. Additionally, the second method on the left there are what are called bee bowls. They are traps with 3 different colored cups placed near the flowers, and it basically attracts insects by color. So they are attracted to either the white, the yellow, the blue, or a combination of both. Essentially, they are filled with water and Dawn dish soap, which traps the insect and effectively kills them. And I sampled these every 48 hours, 3 times during the flowering season as well. And I've just begun sorting through all my insect collections, which has been quite tedious, but I hope that I can do a comparative study between the what I collected in the different methods to see if there's a difference in what I collected. So here's a photo of the inside of a malaise trap collection bottle after just 1 week at the site. This was at the national park, and you can see anything from wasps to moths to carpenter bees and syrphid flies. You can see everything there, and I'm excited to get into it and try to identify what species are at each site. So broader impacts. So why do we care about studying this? So it can help our understanding of how invasive plants are disrupting native ecosystems. Stealing pollinators from native plants can weaken their reproductive success. In this photo I took here at the national park, you can see on the left, there are multiflora rose in total bloom. And on the right is the native Allegheny blackberry, also in bloom at the exact same time. They're both nearly similar looking white flowers that are competing for the same pollinators. So that's why my study is critical for conservation planning and can help protect native plant communities and support pollinator health and ultimately guide smarter restoration strategies. So some special thanks to my advisor, Dr. Oscar Rocha, my lab mate, Elias, for his help, Asiano and John for their help with this experiment, my husband, Donovan, for his contributions to the experiment, and Sydney for her photography. So I guess I'll take any questions that you guys might have.
Awesome. Thank you so much, Abby. Another fascinating presentation. So as people start getting questions in the chat, I have one. So I keep thinking about that jar of bugs that you just showed us. And I remember thinking back to my time in graduate school, and identifying fungi by morphology was like the bane of my existence. So how are you going to go about doing this? Is there sequencing to know what bugs you're looking at, or is it all morphology and you sit there with a huge book and a microscope and you go to town? Yeah.
So right now I've been sorting by order to try to narrow it down slightly. And I realize I'm not an insect expert, so I'm definitely going to have the help of outside helpers. I do have a contact at Ohio State who will probably likely help me figure out what I'm looking at. At this time, I'm not too confident in my abilities to identify the species level for a lot of the flies specifically.
Mm-hmm.
So I'm definitely going to have helpers.
Yeah, it's not something you think about until you start doing it, how difficult it is to identify insects and small things like that.
Yes.
So taking a step back for a second, so you were speaking a lot about how invasive plants can outcompete natives. Can you quickly address why it's so important for ecosystem health that the natives prevail in these types of systems?
Yeah, so I'm really passionate about protecting native plants and basically protecting biodiversity. I don't want an open field of just one species, multiflora rose. That's kind of boring and sad. Like, we want all the different types of beautiful wildflowers that we can find in the prairie or in the forest. And if we allow these invasive species to take over, it effectively like eliminates all the beautiful different types of flowers that I really am passionate about, which is why I care so much about ultimately trying to, end multiflora rose.
Yeah.
Yeah.
Yeah. It's also important, I think, for soil health, right? To have multiple different, and also the other pollinators around, like it increases biodiversity in both aspects. So yeah, but I agree with you. I'm an ornamentals person. We want all of the flowers. So
yes, 100%.
yeah. So many of our audience members are likely really passionate about this topic of invasive plant removal as well, but manual removal of these can sometimes feel pointless because of how quickly they grow back. So do you have any insight on what ways work to help native forests prevail? And also, how does your research kind of connect to these solutions?
Sure. So, I mean, people have tried, cutting and then applying herbicide, but these plants have proven over and over again that that's not effective. And there's really nothing effective other than one thing I observed was that there is this virus called rose rosette disease that I found at 2 of the 3 of my sites. So at the national park, about half the plants were infected with this rose rosette virus. And it basically has these microscopic mites that will feed into the tissue of the plant, and then basically they transfer the virus, and it's basically killing almost half of the field in question that I've showed you, which I was not expecting to notice that it was actually effectively killing them within the last 2 years or so. So I would say that the rose rosette disease is the only thing I know of that has effectively killed multiflora rose. But I know other people have problems with it killing their ornamental roses at their homes or in gardens. So it's not really, the best for mass releasing it on a larger scale, because it will kill other roses too.
Yeah.
So
interesting. Yeah, I had a question queued up for later. I think certain species or varieties of verticillium wilt have been found to reduce the trees of heaven, which is another invasive plant. Yeah. So yeah, I was curious about that, but rose rosette is pretty devastating to other roses in the area. So yeah, might be something to look into later. We had another question about strategies for controlling invasive plants. How do controlled burns work to kind of clear fields, and does that work for multiflora rose?
So basically it can resprout. So you might be burning what's on top, but with the asexual reproduction, it's just going to grow back. And burning's not necessarily going to get rid of the seeds that are still in the soil. So they will keep germinating for that 10 to 20 years. And so fire is not going to be an effective long-term solution, especially since they invade forests. So that might be a problem too with trying to do a controlled burn in the forest.
Yeah. Okay. So we have another question asking about if declines of invasive plants can also be an indicator of overall ecological decline. And is this something that you use as kind of an indicator for that?
So you said decline of invasives?
Yeah. Is that always a good thing, or is it an indicator also of ecological decline potentially?
Ooh, that's a good question. I guess it just depends, right? Like if there is a decline, I want to know what is causing it, what specifically is making that species decline. It depends, right? Is that also affecting the pollinators, hurting the other native plants? I would say it just depends. Like with the rose rosette disease, if it, effectively kills multiflora rose, well, is it also killing the swamp rose nearby that's native? at my one site. So I don't know. It just depends.